Welcome to my lab. Today I'm going to be showing you how to use a microscope to view bacteria. So I'm going to be teaching you how to make a wet mount on a slide of your bacteria and how to view it using the microscope. The microscope I'll be using today, it's a compound microscope, and a compound microscope basically means that there are two different lenses that you look through, but I'm going to explain that in a second. So the basic working of a microscope is that there's a light that shines through your object and then is magnified by different magnifying glasses so that you can see things that you wouldn't normally be able to see with your naked eye. So this is the microscope here. This is the on switch. This is the light, so if I turn it on, you'll see the light come on. So this is where the light is. This is the stage, and this is where you will put your microscope slide with whatever you want to look at. Today we're looking at bacteria, so I'll show you the slide in a minute. These things here are called the objectives, and this is the first set of magnifying that the object will go through. So there are 4 different objectives. All of them have different magnifications. This is the lowest one. This is times 4. This magnifies your object times 10. This is times 40. And finally, this is the big one. This is times 100. Up here is your eyepiece where you look through, and again, this is another magnifying glass. This is why it's a compound microscope, because you have one set of magnification here and another set here. So this is times 10. So whatever you have magnification, if this is times 4 and this is times 10, then you're viewing the objects at times 40 magnification. Other parts of the machine that you need to know, I already showed you the on switch. This controls the amount of light that you get by opening and closing the aperture. So as you can see, if I turn it down, there's almost no light, and when I turn it up, the light gets very bright. So you can adjust this based on the color of whatever you're looking at, whether or not it's see-through. Depending, to see better, you might need to adjust the light. This is what we call the coarse focus, this knob here on the outside, and this is the fine focus. Some microscopes have these as 2 different dials, so one for coarse and one for fine, but for this one, the outside one is your coarse focus and this is the fine focus. And then this knob here, it basically controls the movement of the stage. The stage is this entire thing up here. This is where your slide will go with your bacteria. So this will move the stage. This is 2 knobs, so this one will move it forward and backward, and this knob moves the holder from side to side, so your slide will move. So I'm going to show you how to make a wet mount and how to view it. Wet mount from a bacterial plate. What you're going to need is your microscope slide, which is this, a cover slip, which is this. You're going to be working on the aseptic condition, so you're going to need a flame, a Bunsen burner, your plate with your bacteria on it. This is my bacteria that I'm going to be looking at today, an inoculating loop, and some sterile distilled water. So the first thing, of course, as always, you sterilize your loop by heating it from the stem all the way up. And you allow that to cool. In the meantime, you want to put a drop of water on your microscope slide. So with your distilled water, very carefully, just a drop, just like that. And once your loop is cool, what you're going to do is you're going to scrape the surface of your bacteria with the loop and just put it in the water, and that's how you make a wet mount. So this is cool now. So here I go, I scrape up from the surface of my bacteria, and then it goes here, and I mix it out a little bit so that I don't really want big clumps of bacteria, because those are harder to look at. So you want to kind of spread it out a bit, not too much though. And when you're done, you take your cover slip. Be very careful with these, they can break easily, and you just put it right on top. You try to avoid getting any water bubbles, stuff like that, because again, those will mess with your visibility under the microscope, and that's it. You just sterilize your loop to finish off, and you're ready for the microscope. I have my wet mount ready. I am ready to look at it. So again, turn on the microscope, and what you want to do is you want to bring the stage down to the lowest that it can go. So you can use, what the coarse focus does is it brings the stage up and down, closer to the objective and further away. So you bring it as far down as possible, and you put your slide on the stage. This little arm is to hold it in place. So just like that, and it will snap into place, and then you can move the stage around. So what you want to do is make sure that the part of the slide with the bacteria is directly over the light, and you start with the objective at the lowest level always, and you start focusing using the coarse focus, so you don't touch the fine focus yet. So what you do is you look through here. This is a bifocal microscope, which means it has these two eyepieces, and you look through both of them. It's really hard. Sometimes you're tempted to look through one, but what you want to do is you really look through both of them, and you start to focus. It's important when you're learning to focus the microscope and to see your object is that you want to go slowly and gradually. So what you're looking for, you're trying to bring the object into the best focus, the best way that you can see it. So the first thing you do, you adjust the eyepiece to make sure that it fits your eye and you're seeing through both lenses. So you should be seeing one big circle, not two or 1.5 or anything like that. And then you bring, just with the coarse focus only, you bring the stage up until you can see anything. So an outline of an object, you're looking for bacteria, looking for the shape of your cell, anything at all. Once you start to see it, you pause and you move the slide around to make sure that it's in an area that you can see the best that you can. Play with the coarse focus some more until you are seeing it as clearly as you can at this objective. Only when you've done that, only when you can't, when you know it's not fuzzy either way or one direction or the other, then you move to the next level objective. So I'm going to move from ×4 to ×10, and as you can see, I don't know if you can see, right, so the bigger objectives go closer and closer to the slide. This is why you go slowly and carefully, because you don't want to end up breaking the slide or the objective because you're trying to focus too fast and you put too much pressure on it. So again, same thing with the coarse focus. As you go up in the objectives, you'll have to do less and less, of course, to adjust the focus. This is why you start from the bottom up. Again, move your slide around, make sure it's in an area that you can see whatever you're looking for. I'm looking for bacteria, so yes, I am seeing some areas that look like they have some cells. So I just play around with the coarse focus until I'm seeing it as clearly as I can. Again, you move to the next objective, which is a ×100. Once you get to this objective, you start to use the fine focus. So you stay away from the coarse focus. And that's because your slide is really close to the objective. So if you bring the stage up any further, you risk breaking the slide. So again, this objective, you just use the fine focus. You can move the coarse focus very carefully. I wouldn't recommend you do this until you have some experience with, gauging the focus. And you use your fine focus. I'm going to play around with my light a little bit because my cells are a bit dark. So I'm just going to adjust the light here so I can, oh yeah, there we go. See them a bit better. And looking around my slide. So what you want to do is scan your slide because you might think, hey, this is a good patch of bacterial growth. And then you move over on your slide a little bit and you see a better patch. So you want to move around a little bit until you're seeing what it is you're looking for and the best possible picture that you can get. And once we focus at this objective, we move to the next one. But the ×100 objective is used at oil immersion. What oil immersion is, it means that because the objective is so close to the stage at this point, it's going to be touching the slide and you don't want anything to scratch or to break. So what we do is we apply a drop of oil. This is the oil that we use on top of the slide before we put the objective down. So what I've done, I've twisted the objective so it's in between the ×40 that I'm just coming from and the ×100. And with my bottle of oil, I'm just gonna add a drop right in the middle on top of my slide. just a drop, one drop will do it. And then I'm gonna twist my objective very slowly and carefully so that it's on top. It's basically the same at the other stages, except you absolutely do not adjust your coarse focus. You can't bring the stage up anymore. It's literally touching the slide, you will break everything. So you simply use the fine focus to adjust it the best that you can. You can move the stage around a bit at this point until you see your bacteria. So there we go, I'm seeing some cells. There we go. Use your phone to take a picture of your slide. It's really hard to do on video, but I'm going to try to show you. I don't know if it will work. But the idea is that you put the focus of your phone into the eyepiece. You're going for this circle. So what happens is the phone gets really weird at this point. It works a lot better off video. So what I'll do, I'll take a picture and I'll upload it. Oh, there we go. That's kind of it. So you can see some cells in there, definitely, right? So that's the idea. The slide from the microscope when you're done is just as important as when you put it on, because there are a couple of things that you have to remember just for the care of the microscope. So if you have it on oil immersion like I do, the thing that you want to do first is to move the stage down. So you use the coarse focus and you move the stage down. Once you've done that, you turn off the light. You shift it to the lowest objective, which is the ×4. That's what you always leave the microscope on. You pull the lever and remove your slide, and you plug out your machine, and that's it. You if there's dust on the microscope, you know you want to wipe it off. If your lenses get dirty, you use lens paper to clean them, because you will end up with fuzzy images if you don't. Just like a camera, you have to clean the camera lens, you have to clean the microscope lens. And that is it for the use of the microscope. I hope you learned something today, and I'll see you at the next tutorial.